السلام عليكم ورحمة الله. أتمنى أن تكون الجميع بخير. Today we have comparatively a small and easy practical. It is mainly about parasitic helminths. I will try to keep it very short and precise. As you know, a couple of parasites are found in the intestine of man. Among them is Enterobus vermicularis, which is the pinworm. It is called pinworm because of the shape. As you see in the center, the elongated worm. It starts, for example, from the upper side, showing a tubular or muscular esophagus, and then it has a glandular, this bulb-like structure portion, and from there, a long tube going through the body, ending almost near the anterior end. What you see in the middle, which is taking dark structures, these are uteri, or the uterus full of eggs. And the eggs, they have characteristic shape, as you see down here. They have somewhat convex side and one straight side, and usually you will see the developing embryo, which is the small new larva. So usually this parasite causes infection by ingestion of eggs, by inhalation of eggs, and sometimes by what we call autoinfection, because at night the female crawls to the preanal skin and it lays eggs there, and the eggs can hatch within 4 to 6 hours, and they can crawl back, causing autoinfection. This parasite is very common among children because of their unhygienic behavior. To the upper right, you will see the parasite with a curved end. This is the male, which is typical for many of the male nematodes. They have curved tails. If you see this diagram, the upper right parasite it has comparatively short and stout posterior end and a very long and thin anterior end, so it looks like a whip, and that's why the name whipworm. They are known as Trichuris trichiura, and they have, of course, the male, which is to the left lower side and ending with a posterior tail or posterior end. Down to your left and right are the eggs of Trichuris trichiura, which is very characteristic. It has thick eggshell and 2 lateral plaques. The one to your left is stained, and the other one to the right is just recovered from iodine solution, so our preparation in iodine. Ascaris lumbricoides is known as the giant roundworm. It could be up to 30 centimeters long, and the down one is the male, which is thinner and smaller. It has curved posterior tail, and the female is larger because it has to produce a lot of eggs. They say Ascaris produces about 200,000 eggs per day. That's numerous one. In the lab, you could see different forms of Ascaris egg, but the typical ones which usually you see are the one to the lower left, which is showing thick albuminous mammillated eggshell. The one, for example, just to the right, it under little pressure in a technique known as cat-to-cat technique. So it is also a fertilized egg. The one to your right after that with a smooth eggshell, we call it decorticated because it lost the albuminous mammillated eggshell. And the one to your right, which is barrel-shaped, is unfertilized Ascaris egg. So the common egg which usually you see are the two ones to your left. We have a couple of parasites that cause infection by the penetration of their larvae, and these are Ankylostoma duodenale and Nicteria americanus, which we call the hookworms, and the smallest nematode infecting man known as the Strongyloides styrocoralis. So the larvae stage known as filariform larva. So these, they penetrate the human skin, and they are carried by the blood. They go through the liver, then to the heart, and then they settle in the lung. They develop and then coughed up and swallowed to be adult parasite in the intestine. The eggs of Ankylostoma and Nicteria, they are very typical and indistinguishable, and you could see the one here, it is showing very thin eggshell. So if you are suspecting hookworm egg, please reduce the illumination or the light of your microscope. If it is too strong light, it will be difficult to see the eggshell, but you see those developing morula of cells. If you look to the males to your right, it has somewhat a sort of an umbrella sort of tail. It is not curved, but inflated into an umbrella structure. Some chitinized structures connected together with very thin membrane, so they have a shape of an umbrella. And people, they say, usually it may be used during copulation. The male holds the female by this copulatory bursa. It is called copulatory bursa. The eggs of hookworms are passed in stool, and then they hatch to release the larva. This larva is known as rhabditiform larva. It's a non-infectious, it is a free-living larva. After 2 to 3 days, it molts to become an infective filariform larva. If it comes in contact with our skin, it penetrates, and then it is carried through the liver to the heart, and then in the lung, and then coughed up. So, of course, when they're going through these organs, these larvae, if they are in big numbers, they can cause some, for example, hepatitis in the liver, and of course, they can cause pneumonitis in the lung. Strongyloids larva is also, that is a free living, but it transforms into an infective filariform larva that just cause infection like ascaris by skin penetration. However, this parasite can cause what is known as disseminated or hyperinfections, especially among immunocompromised individuals, because usually the eggs of this parasite hatch inside the intestine, and from there they can cause internal autoinfection. And if they come to the preanal skin, they can also cause external autoinfection. So precisely, strongyloids, especially among immunocompromised individuals, they can cause disseminated infection, or what we call hyperinfection, because it is capable of causing a skin penetration and causing internal as well as external autoinfections. We have a couple of filarial worms. Among them is Onchocerca volvulus, causing river blindness, and it is transmitted by the bite of what is known as the black fly, Simulium damnosum, which breeds in fast-running rivers. Usually, these filarial worms, all of them, they have typical life cycle, except the insect vector is different. So precisely, each one is transmitted by an insect, and instead of giving ova, the ova are produced inside the uterus, but they have to give a moving stage known as microflare. And excuse me, to the lower right, you could see a diagram of a microflare, and if you look to the end of this microflare, which is drawn diagrammatically, there is almost just an extension showing what is known as the sheath. So 3 species they have sheath microflare, and these are Wucherella bancrofti, loa, and Onchocerca volvulus. The other ones, they don't have sheaths. These many small dots are known as nuclei, and the distribution and how they are ending in pairs or just up to the tip or before that could be helpful to identify the different species of filarial worms. Onchocerca volvulus, usually it forms what is known as sarcomas or nodules, usually on bony areas, and if it is on the head, of course, the microflare can migrate and they can cause eye problem, leading from somewhat partial or sometimes even complete blindness. This is Wucherella bancrofti microflare. It is just a snake-like structure with plenty of nuclei, and the arrow shows the end of the free part of the sheath, which could be seen at the anterior and posterior end, because it is a sheath which is fitting the body, except could be seen at the anterior and posterior end of this stage. Trichinella spiralis larva in the muscles, because trichinella causes infection when people eat undercooked pork containing the encysted larvae. The encysted larvae will be released in the intestine, and then they become adult parasite, and it releases larvae which can enter the muscle, especially striated muscles, as you can see in this photograph. You see this is a larva or larvae in what is known as nerve cells, and usually this is taking place, as you can see the striations, so it is found in striated muscles. It survives for some time, and sometimes it dies and calcifies. Two important tapeworms which are common. These are Tenosolium, known as the pork tapeworm, and the other one is Tinea saginata, which is known as the beef tapeworm. People acquire beef tapeworm by eating undercooked beef from cattle. Tenosolium is the pork one. This is the scolex, showing 4 suckers, and rostellum, which is armed with 2 rows of hooks. So these 4 suckers and the rostellum and the hooks are used for attachment to the intestine. It may be easy if you have stained proglottids or segment to identify Tinea saginata gravid proglottid from Tenosolium by the number of uterine branches. What you see here are the uterus is highly branched in both segments. So if you count 25 or 30 latrine branches, this is typical for saginata, and if it is about 15 or less, this is typical for Tenosolium. It is difficult to distinguish the Tinea eggs. So we have what is known as Tinea species eggs. This typical egg could be an egg of Tinea saginata, of Tenosolium, or Echinococcus species. And we have a couple of Echinococcus species causing hydatid disease, and their eggs looks like Tinea eggs. So we call it Tinea species and Echinococcus egg. All are characterized by being circular or rounded with thick striated eggshell, and internally we'll find the hexagons embryo, the embryo with 6 spines or hooks. And so in the intestine, for example, if ingested, the eggshell will break and the small parasite will go out of the intestine to form, for example, in animals, the bladder worms or cysticercus stages, which are eaten in undercooked beef or pork, and they can cause infection in man as other parasites in the intestine. However, Echinococcus egg, if ingested by man, they don't become adult parasite, but the egg will leave the intestine to form what is known as hydatid cyst anywhere in the body, but mostly in the liver, because they breaks through the hepatic portal vein, and this is going through the liver, and that's why they cause most of those bladders in the liver, but they can cause also anywhere in the heart or even in the brain, as well as, for example, other organs and tissues, and sometimes even inside bone marrows, long-shaft bone. This is an adult Echinococcus granulosus. It is common among people who are breeding, for example, cattle, and they're having, or sheep, and they're having dogs to help them in controlling those animals. So people who are living in close contact with their dogs are more at risk, or are at more high risk with this infection. The parasite is simple in structure. It has a scolex with 4 suckers and rostellum with hooks, then a segment which is young, a segment where you see male and female reproductive system. This is mature one, and the last one, usually the male and female systems, reproductive system disintegrate, and the uterus will enlarge to fill almost the whole segment full of eggs. The hydatid cyst is almost a fluid-filled vesicle or bag, and the internal layer of it is capable of budding out and even forming daughter cyst, and the daughter cyst can also bud internally. So one cyst may be containing many scoleces, and here you could see, for example, it has comparatively thick, and then many going through many striated muscles, and then it is here. Of course, surgery of hydatid cyst has to be very careful surgery. You may need to aspirate so that you don't, if it is broken, then the body will be flushed with the fluid containing many scoleces. So it has to be a very careful surgery. You may need to aspirate, and you may need to inject, for example, some material to kill the scoleces and then to excise the cyst. The dwarf tapeworm is called dwarf because it is small in size. It has characteristic scolex, which is 4 suckers, rostellum, and hooks. The egg is comparatively a large egg with large egg shell, but it is a light one, not striated, and you will see the internal part is the hexacon's embryo. The egg also may be characterized to have almost filament-like structures at the polar end in the egg shell. Schistosoma, this is a scanning electron micrograph to your excuse me, to your left, and you can see the oral sucker and ventral sucker of the male. Behind it is part of the female, which is carried in the gynecophoric groove. It is a longitudinal folding of the male to carry the female there. So usually people are infected with schistosomes if they bathe or they swim or they wash in water containing the snail intermediate host of those parasites. The snails will be infected and they release swimming stages known as sarcaria. So if the sarcaria comes in contact with the skin, it penetrates. Usually the tail of the sarcaria, as you see later, is broken out and the anterior end is becoming almost elongated or ovular, and it has rough surface. It keeps wriggling in the dermis epidermis layer of the skin, and if it is not killed at that stage, then it enters the circulation and will be carrying up to the liver. They become adult parasite, and then they migrate after copulation to reside in the mesenteric veins in case of Schistosoma sonai japonicum, or in the vesical veins around the urinary bladder in case of hematobium, and they can survive for up to 30 years. To your right, these are just stained male and female schistosomes in copula. It is easy to distinguish between the three species, important species infecting man, by their eggs. Mansoni is having a lateral egg is having a lateral spine. In hematobium, the spine is terminal, and the shape of the japonicum egg is almost rounded and with a small rudimentary unclear spine. This is sarcaria, which is usually found in water released from the snails, and during the penetration, because it has some salivary glands that produce enzymes that can lyse the tissue, and then it triggers to get inside, and during this process, the tail is cut off, but sometimes it is entered with the tail, and then it's known as schistosomulum, which will develop into another parasite. The intermediate host for schistosomes are snails. For example, this Biomphilaria snail, different species of it, they can carry Schistosoma mansoni. This is Bolanis snail, which are intermediate host for hematobium. And these are Oncomelania hubensis from China, and they are intermediate host for japonicum. You will find some snails somewhat similar to this, but they are more conical, more darker in structure, known as Melanoides tuberculata here in Oman, so they don't have any medical importance, the Melanoides tuberculata. Here, these are fresh water snails from Oman. The upper three ones, A, B, C, are Limnia snails, and Limnia are intermediate host for Fasciola, and we have two important Fasciola species, mainly infecting animals. These are Fasciola hepatica and Fasciola gigantica. Sometimes human infections can happen by these two parasites. The tiny small snails, they are known as Bolinus wrighti, DEF, and usually they are the intermediate host for Schistosoma hematobium. The other two to your right, that is G and H, are Biomphilaria snails. They are called here at the beginning, they are named Biomphilaria arabica, but genetic studies show that they are typical Biomphilaria 5.3, which are very common along the Rift Valley in Africa. So possibly these snails used to be the same species before the continental drift. So that's why it may be coming from Africa before the continental drift. All the other species from I to N, Melanoides tuberculata, they are very popular here in many of the water bodies, whether these are springs or phallic, but they don't have any medical importance. As we know, it's not carrying any parasitic infection of medical importance. Thank you very much. So please do revisit these slides every now and then so that they stick to your mind and you can easily identify these slides if they are presented in the exam, as well as answering one or two relevant questions to the demonstrated photograph in the exam. Good luck. Assalamu alaikum wa rahmatullah. Stay safe.